Hi, Dr. Wang here. Today we're going to go over this assignment, a bioinformatic assignment called APE3. We're going to learn to use APE to simulate a restriction digestion. The results coming from this assignment will also help you interpret the gel image in Lab 4. So let's get started. To complete this assignment, first you will need to use these two sequences coming from APE2. So these two files are located under APE2 annotation. I already have these two files downloaded on my computer, and I have opened one of them with APE. So this is the sequence for PMSH2, the entire plasmid that's more than 9,000 base pair long. So how do we simulate Lab 3, which is a digestion of this plasmid DNA with APA1 and XBA1? To do so, all you have to do is to go to enzymes, then enzyme selector. All right, so and then just simply pick, I actually already picked them, APA1 and XBA1. So there is a parenthesis with a number within. This number tells you how many times does this enzyme cut the plasmid DNA. So after selecting the two enzymes, I'm going to click digest to simulate the restriction digestion. So here we have a simulated gel image. The first lane contained the DNA letter. So this is a series of DNA fragments with known sizes. Lane 2 contained the digested sample. So we can see that there are two fragments, right? So one of the fragments, its size is listed right here under the column for size. One of the fragment is about 4,800 base pair long, a little more than 4,800, and the other one is a little more than 4,400 base pair long. All right. So here, when you click on the fragment on the gel image, the relevant number is already highlighted. Okay, so here's how to do simulated restriction digestion in APE. And by comparing this simulated gel image to the gel image from Lab 4, we will be able to determine which of the three samples, A, B, C, actually contain the MSH2 insert. So in the end, I would like to compare the digestion of a linear DNA molecule with the digestion of a circular DNA molecule. So what we have in the lab is plasmid DNA. So this is a circular DNA. If you cut this twice, right? So with APA1 cut once, right? So XBA1 also cut once. You're going to get two fragments, like so. If you have a linear DNA, if you cut it twice, this is going to give you three fragments, right? So there is a difference depending on whether the starting material is linear or circular. Coming back to APE3, there are a couple of questions in this assignment. One of them relates to the digestion of plasmid with APA1 and XBA1. So when you do simulated digestion with those two enzymes, please save the image and then place those simulated gel image in your notebook. And then there are also other questions in here just as exercises, right? So you can just repeat the step. You can, for example, not one, right? So for not one, all you have to do is to come back to APE. I'm going to close this simulated gel image. By the way, please take a screenshot and place it into your notebook. And then come back to enzyme selector. So here I'm going to click to deselect this two enzyme, and all I have to do is to click not one. And this is going to generate the answer for the first question, and then you can repeat the step for the remaining questions.